Hello friends, and this is Dr. Farheen with the next lecture in medical microbiology, and today we are going to discuss bacterial classification. Bacterial classification. Now, why do we need to do bacterial classification? What are the reasons for bacterial classification? So there are two main important reasons why we need to do bacterial classification. Number one, we need to organize the bacteria which we have isolated or we are studying into groups and species so that we can relay the important information about them to our scientific community. The second one is to lay a criteria for this organization. Yani to describe a bacteria in such a way that based on the specific characteristics, you will be able to characterize them, to able to organize them, give them a specific name, and put them into specific species which belongs to a specific family, and so on. So these are the two important aims of bacterial classification. Now, what are the bacterial classification ways you can do? Bacterial classification can be done in mainly three important ways. Number one, we can do it as a phenotypic classification. Number two, you can do analytical classification. And number three, you can do a genotypic classification. So what is phenotypic classification? Phenotypic classification is based on the morphology. It is based on the morphology and its maybe inner capacity, so on. Mainly the what it has in it, okay? Whereas the analytical classification is based on chemicals present in it, chemical molecules present in it, okay? Maybe it is lipids, proteins, carbohydrates, so on. And genotypic classification is based on the DNA or the genome. It is based on the genome or nucleic acids, genome or the nucleic acids present in it, okay? So now let us see now one by one what is phenotypic classification. Now in the phenotypic classification we can do it in several ways. Main important are 7 types of characteristics which are considered for phenotypic classification. Number one, microscopic morphology. Macroscopic morphology. Number 3, metabolic activity. Number 4, biotyping. Number 5, serotyping. Number 6, antibiogram patterns. Number 7, and the last, phage typing. So let us see one by one what are these in detail. Now coming to the first one, the microscopic morphology. So this you can understand by the thing itself what I have written. It means the morphology of the bacteria which you can see in the microscope. So what is the shape of the bacteria under the microscope? Now under the microscope, the bacteria can be classified into either of the following. Number one, spherical bacteria. Spherical bacteria, yani these are ball shaped, or you can say spherical in shape. These are named as cocci. Rod shaped. Number two, rod shaped or brick shaped bacteria. These are referred as the bacilli. Then the third one, this is number one, this is number two, and number three is comma shaped. Yani they are curved, slightly curved like this, like a comma. These are referred as the vibrios. Then you have another category, number four, the spirals. They are spiral like this. These are known as the spirochetes. Now here I will just have another category which I will refer as others. Yani these are they may be different shaped, like because of certain characteristics they may be shapeless, or they may be they are intracellular bodies, or they may be obligate intracellular parasites, so they may not show a particular type of morphology, so on. We will refer to them one by one when we go into the details of the chapters. Now basically what here I want you to stress on is, in the microscopic morphology there are basically four types of bacteria. That is, either they are spherical in shape, or they may be rods or brick shape, there may be comma shape, or they may be spirals or spring like. The one which are spherical are referred as cocci. The one which are rod shaped are referred as bacilli. The one which are comma shaped are referred as the vibrios, and the spirals or the spring shaped are known as the spirochetes. Okay, I hope this fact is clear with you guys. Now the examples I can give you here, like cocci, you can just remember the example, the most common example, that is the staphylococci. Bacilli, you can just remember E. coli. Example, E. coli. Vibrios, easy to remember. Example, Vibrio cholerae. And spirochetes, example, Treponema. Okay, now let's go to the next part. Macroscopic morphology. What is when we are talking about microbes, why are we talking about macroscopic morphology? Macroscopic, micro, when we say micro it is small, and when we say macro it is big. So what are we meaning by macroscopic morphology? Here microscopic morphology refers to colony morphology. What is a colony now? This term colony refers to a group of bacteria that have grown that have grown on agar that have grown an agar from a single microbe or microbial cell. Okay, when you put bacteria on the agar, when you inoculate them into a agar, they will show specific morphology. Like suppose this is a petri plate, and you have inoculated the bacteria, say using a loop like this, then they will, each bacterial cell is going to grow into a group of cells. It is going to divide, multiply, reproduce, whatever you say, and they are going to produce something like this. So these individual group of cells which have grown from one single microbe are referred as the colony. And by the term macroscopic morphology refers to the morphology of this colony. Now, why is it useful? Why is it needed to study? Now, when you are doing the diagnosis of certain bacteria, you are going to culture the specimen into the petri plate or into the agar, so that you can multiply those organisms which are present and elucidate their characteristics and put them into a specific group. That is, you are going to identify this organism. So when you have grown these microbes on the agar, they give you a particular type of colony, and this colony morphology will give you a hint how to proceed with. Now, most important one, I will give you an example of streptococci. When you have grown them into a agar, the colony may show you, say, hemolysis. It may be hemolysis based, how the bacteria is lysing the RBCs in the agar. So based on the hemolysis, you can classify the colony as alpha hemolytic, beta hemolytic, and gamma hemolytic. What are these individually we'll discuss later. For understanding this, you can wait for the upcoming videos on streptococci, where I will be discussing in this one in the detail. Then you can also have consistency. Based on consistency of the colony, like whether it is a moist colony, or it is a dry colony, or it is mucoid colony, so on. So each one of them will, this is a specific characteristic of particular bacteria. So based on certain characteristics of the colony, you can get a hint which bacteria it might be and what are the specific biochemical tests you need to do for this particular bacteria. So this all we discuss in macroscopic morphology of the bacteria. Now, number three was the metabolic activity. Now, once you are done with the outer morphology of the bacteria, here you are getting inside the morphology of the bacteria. Now, to know what are the things that are present within the bacteria, you need to check for its metabolic activity, whether it has this enzyme so that it produces this. For example, the most important, most common one, say if it has, you want to know whether it has an enzyme to ferment or utilize a particular sugar. Utilize a sugar. Say you want to see the utilization of lactose. Okay. So what you are going to do, you are going to subject the bacteria for the biochemical test, and the results will let you know whether this bacteria has the capacity, the bacteria has the bacteria has the capacity to utilize a particular sugar or not, and that particular characteristics is going to give you a set of biochemical reactions. Say you are going to check for lactose, sucrose, maltose, so on, and here you may get positive reaction, here you may get positive reaction, here you may get negative reaction. Now based on these metabolic activity, you can categorize a bacteria, and hence you can name them or organize them under a specific group or the species. Clear? Now going to the next one is the biotyping. Now what is biotyping? Biotyping is nothing but a specific group is subdivided into several other groups or subgroups based on certain particular type of characteristics, like maybe based on the toxins they produce. The bacteria produce a certain toxins. So based on whether particular toxin is produced by the bacteria or not, you can biotype the group. For example, you have E. coli species. You have E. coli species, and if it produces a particular type of toxin, it can be biotyped as a certain particular group. For example, O157. This is one of them. Or you can also have Shigella, so on. So basically what here important one thing you have to remember is biotyping is based on one specific character where a particular species is subdivided into other groups or subspecies type. Okay. Now this is usually done in the reference laboratories. We usually do not do in a regular basis. The next one is the serotyping. This is based on, now sero refers to serology, that is mainly the contents are present in the serum. So serotyping means the bacteria is classified based on antigens. It has sometimes the bacteria are inert. They do not show much of the biochemical activity. So here you are going to rely upon antigens. A particular antigen, especially in the Salmonella group of bacteria. You are going to look for antigens present on the bacterial cell, and you are going to name them and organize them, or you can say you will be able to identify them. Then the sixth one is the antibiogram. Patterns. What is antibiogram? Antibiogram is the sensitivity pattern. Antibiogram is the sensitivity pattern. Antibiotic sensitivity pattern. Now what is antibiotic sensitivity pattern? So in the lab, what you are going to do, you are going to test the bacteria for sensitivity to different antibiotics, and there you are going to get, like say Escherichia coli, which is sensitive to say here this particular pattern, like a particular bacteria is sensitive to certain antibiotics, that gives you the antibiogram. So you can have the, like, most important here is there are beta lactamase resistant or sensitive bacteria. So you can classify them like this, or you can say methicillin resistant staphylococcus aureus or so on. So this particular type of criteria is based on the sensitivity pattern it shows to several drugs. Finally, we have phage typing. Phage typing. What is a phage? What is a phage here? Phage refers to the virus. Phage refers to the virus that infects the bacteria. Yani this is the one which specifically bacteria is a host. The virus which infects, which develops into the bacteria. So there are several viruses which can infect certain bacteria. So based on which bacteria is infected by particular type of virus, it can be phage typed. This is again done in the reference labs. So the most important one, which is important for our diagnostic purposes or on a routine day-to-day life, is the microscopic morphology, macroscopic morphology, metabolic activity, and antibiogram patterns. These are the most important ones what we need. Whereas biotyping, serotyping, sometimes they may be useful, whereas phage typing, even though it is useful, it is mostly used in the reference laboratories for epidemiological studies to see in which part of the world which bacteria is most common. Now, coming to the next one, the analytical classification. Analytical classification is based on the chemicals present in the cell, chemicals present in the bacterial cell. Say you can just look for what is the percentage of proteins, you can look for percentage of lipids, you can look for percentage of carbohydrates. What is carbohydrates? So based on this, you can classify the bacteria based on analysis. Now, usually we do not do analytical classification in the routine diagnosis. Why? Because it is labor intensive, and the instruments needed for analytical classification are very expensive. So the cost effectivity of this classification is very high. So we usually do not do it, and it is done mainly in the reference laboratories. Now, the third one is the genotypic classification. Genotypic classification. What is genotypic classification? Genotypic classification is mainly based on the nucleic acids, nucleic acid content. Here you are going to isolate the nucleic acid. First step is you are going to isolate the nucleic acid, then analyze the nucleic acid. Now, even though this particular thing is costly, nowadays this has developed in a very advanced manner, and it is helpful in rapid diagnosis of several diseases, like in the recent recent pandemic which is still going on. You can see the diagnosis of COVID-19 is done based on the nucleic acids of the virus. Now here this can be done in several ways, like you can have example, You can do nucleic acid hybridization. You can do nucleic acid hybridization, or you can do the PCRs, say it may be RT-PCR, so on. So this is mainly based on the analysis of nucleic acid. That's what is important. Okay, so genotypic classification is based mainly on the analysis of the nucleic acid, mainly the DNA. Okay, that's all about the genetic classification. So these are the most important things which are included in the lecture bacterial classification button, and hit the subscription button to get notified with new videos which are uploaded in this channel in the lecture series of medical microbiology. Soon I will be posting the lecture on bacterial cell morphology. Thank you guys. Thank you for listening.